So I wanted to make this video about viruses, virions, viroids, and prions just as a really sort of quick summary. What are the important points to remember? And the first most important point to remember, all of these are extremely small. And in some books, they're called filterable because cells are actually much larger than this. These do not have cells. These are acellular pathogens. It means that they do not have cells. A means without. Cellular, of course, is referring to the cell. They do not have cells. They're all pathogens as well. They don't live on their own. They have to go into a host cell in order to have any sort of life processes like replication. So they're absolutely extremely small so that they can fit inside of other cells. And so this is a really kind of interesting concept because there's actually a lot of debate regarding whether or not we can consider these alive. And if you consider that for a second, it's like, well, they can't exist outside of other cells. They can't replicate on their own. Obviously, that means that they do not fit the biological definition of life, which is that it's able to reproduce. However, viruses have the ability to evolve. They have evolved huge specificity along with their hosts. They have evolved their genetic code to specifically help them replicate by using host cells machinery. So maybe they are alive because they just, even though they don't have their own machinery, they use somebody else, they use another cell's machinery, basically. So that's a huge debate in science. Viruses, in terms of structure, they're not because they don't have cells. They're classified by their structure and by the genetic codes that they have. And so let's talk about the structure for a really quick minute here. So the structure, the way that I remember it, the parts of a virus that are absolutely essential, is bad news wrapped in a protein. Okay, so that's, someone else said that, I didn't come up with that, in order to kind of describe what's going on with proteins, with viruses. Okay, so I'm just going to draw just a simple virus over here. Okay, so bad news wrapped in a protein, that's the simplest structure you could possibly think of, where the bad news is some type of nucleic acid. And it's either going to be DNA or RNA. It will never be both. And then the protein that's referring to, it's wrapped in a protein, that's the capsid. And that's made of proteins called capsomeres. So that's the, essentially, there are some viruses that's all they have, so we consider that the minimum that you need in order to become a virus, in order to be classified as a virus. So this DNA has information on how to make the capsid, and then it copies itself, and it's able to take over host cells. Some viruses have an additional fatty acid layer on the outside, like a membrane, called the envelope. It's made of fatty acids. This is different than a cell membrane because there is no need for selective permeability here. These don't eat. They don't have metabolism. They don't have cytoplasm. There's no toxins here that need to be released. There's no food that needs to come in. So this fatty acid membrane is just kind of like an additional layer of protection. But we can also destroy those enveloped viruses a little bit easier than we can destroy the protein capsomeres themselves. So if it has to have an envelope, likely that's got some sort of recognition on the outside that recognizes the host cells for attachment. And so if we eliminate the envelope, we can eliminate the attachment and totally get rid of it. So things like hand sanitizer, which have alcohol dehydrates fatty acids. So that's one way that we can get rid of those types of viruses. So the envelope virus is a little bit easier to get rid of. However, if it doesn't have an envelope, it's a little bit more challenging because you're going to have to maybe apply heat to get rid of those proteins that are there. Okay, so that's viruses. And whenever we have a complete viral particle, because we cannot call it a cell, so we call it particle. So whenever we have a complete viral particle, we can call that the virion. So the virus itself, it could be just this nucleic acid in the cell, and you can imagine that if it's a DNA virus, then that's replication is going to take place in the nucleus, or if it's an RNA virus, its replication is going to take place in the cytoplasm, so we can correctly call that a virus because it's just part of it. And the full complete particle that is ready to infect, including at least the capsid, an envelope if it has it, spikes if it has it, all that sort of stuff, that's called the virion. So that's the difference between viruses and virions. Okay, so I want to briefly talk about viroids and prions next. So, viroids. So these viroids, name looks very similar to virions. So these viroids, they mostly affect plants. Their genomes are extremely small and made entirely of RNA. So if we were to draw a picture of the DNA of maybe like a bacteriophage, maybe it would be very large like this. By contrast, the viroid DNA would be like this. Oh, sorry, viroid RNA. Okay, so these are very specific to plants. That doesn't mean that humans don't have to worry about them. They affect crop harvests, they affect like potatoes and tomatoes and those sorts of things and make them grow much smaller. And so this teeny tiny piece of DNA, or sorry, rather RNA, this tiny nucleic acid causes a whole mess of trouble. So the interesting part about viroids is that they have no protein component. They are just the RNA. So it's this teeny tiny little ribonucleotide sequence that's going and causing all of this trouble, especially in plants. And the last category I want to talk about are the prions. So these are proteinaceous infective agents that have no nucleic acid component. So that means no DNA and no RNA. And this is troubling because how do they replicate? If they do not have nucleic acids, how do they replicate? And this is particularly important because these cause a lot of diseases like mad cow disease, links with Alzheimer's, Kuru, a lot of like spongiform encephalopathies, variant Creutzfeldt-Jacobs syndromes, those types of things. And so basically what happens in prions is that these are misfoldings in the protein structure. And so maybe they start off with like a lot of alpha helices in their tertiary structure and maybe just one beta strand. And then something happens, the mechanism of which we don't fully understand, and then instead of having all of these alpha helices, maybe we just have a couple, and then it goes mostly to beta strands. So this new form causes all sorts of trouble. So this new structure, through an unknown mechanism, makes these very difficult to stop. And then once one starts misfolding and causing all sorts of trouble, then it causes others around it to misfold and cause trouble. And so the exact mode of this infection is not particularly known. Is it like what particular agent is causing this? It causes all these devastating diseases. All of these affect, the brain tissue, that sort of thing is also very difficult to diagnose because you have to actually physically look at the brain tissue in order to identify that this protein misfolding is really what's going on. Also, prions are extremely difficult to eradicate. A lot of them can withstand autoclaving. So you have to get particular prionzymes in order to eliminate all prions from any equipment that you're using during an autopsy, or if you suspect any sort of prion, you have to be very careful in how you're cleaning it up, because autoclaving itself will not do this. Rubbing your hands with alcohol are not going to protect you. Gloves, like the regular nitrile gloves, these particles are so small they might be able to penetrate through. So you have to have extra protections, extra cautions anytime you're involving a proteinaceous infective agent. It has no nucleic acid, mechanisms unknown. There have been a lot of strides working towards understanding this further, but we are not quite there yet. Okay, so to summarize, our viruses, those are their bad news wrapped in the proteins. The virions, those are the complete viral particles. The viroids, there's a small RNA that attack plants mostly, and the prions are a proteinaceous infective agent. They're really difficult to get rid of, and a lot of them cause spongiform encephalopathies. They're all extremely small, tiny. There's a debate whether or not they're alive. They have similar structures in that they don't have cells, and also in many cases, especially for the viruses and virions, they are very specific in what they attack. So for example, the viroids only attack plants. The prions for the most part attack the brain tissue. Viruses can attack specifically liver cells. They can attack specifically birds, they can attack specifically humans, etc., etc. So they all share a level of specificity as well, despite not being technically alive by the biological definition. But if you look at them and consider that they are evolvable, that is debatable. Okay, so thank you for your time.